Okay, this is a recording for morphology, transport, and I'll throw a little bit of plant response in there as well. So we're going to start off with the basics here. We'll talk about roots, shoots, and leaves. The first part, roots, we need to know what the purpose of a root is. Right? So the purpose of a root is threefold. One, and the primary, is to absorb. Right, so we're looking for absorption of water and minerals that are found in the soil so that the plant can use those for other metabolic purposes. The other thing is to anchor the plant in place. So we see a difference in monocots versus dicots. Dicots having more of a taproot and monocots having more of fibrous root structures. And then the last one is for storage. And that comes with things like carrots, right, where you have this gigantic large taproot that could store a lot of energy, and even if the plant gets clipped off for some reason, shape, or form, that root can give enough energy for the plant to revive itself and regrow after the damage that has been done. Or from winter to spring, so enough energy can be passed up. Alright, so the next part is looking at the root structure. So here we have a picture. This showing a dicot. The reason I say it's dicot, two reasons. One, in this diagram here, this picture, you can see that the venation of the leaves is more spread out, so that's a characteristic of a dicot. But the big thing here, when I look at a crosscut of a root, I see that the phloem and xylem are making this X-shaped pattern inside the stele here, and that is characteristic of a dicot. If you saw a monocot, that stele, you would see the xylem and phloem more kind of spread out, almost in a ring formation inside the stele. Right? So if this was a monocot, you'd see this center blue section looking more like this in a monocot. All right, so the root, we said absorb. So with the absorb, we've got the three layers of tissue. So we've got the dermal layer, which is going to be the outside. You've got the ground, which is going to be everything between the dermal and the vascular, and then you've got the vascular tissue, which we've already talked about here, which is called the stele in the root. So all the blue stuff in there, this is all vascular. The brown stuff around the outside is dermal, and the red and orange tissue is going to be your ground tissue. You can see in the root, the purpose of the dermal tissue here is to actually branch out and start making these finger-like projections called root hairs. The whole purpose of those root hairs is for surface area. A common theme throughout all of the different sections that we've talked about, surface area is all about increasing the ability for absorption. Right? So we see it in the human lungs with alveoli, we see it with gills, but we're not looking to absorb water in those locations, we're looking to absorb gases. We see it in the small intestine for absorbing all of your little monomers that you have broken down via the digestive process. All right, so the next parts of the root, you look at the end part of a root, we're going to see meristematic tissue. And since it's at the tip here, it's going to be an apical meristem. The apical meristem is going to do mitosis. So we see at the very end of the root is where apical meristems are found, or the very end of a shoot, which we can talk about later, is where we're going to find apical meristems. So we get the division here. We get this root cap, which is going to be protective because you're pushing through a hard substrate. You do not want to destroy this root, so there's all these dead cells that have some extra lignified properties and cell walls that help protect it. And then you have the zone of elongation, which is right behind the meristematic tissue, so we get all the growth back there. So divide the cells, protect the cells, and then that elongation, that root can then push down through the soil. So that is the root. The next part is the shoots. So like I said, at the tops of the shoots, we have the apical meristems. We're going to get a lot of mitosis, and that's where the plant is going to grow. And we have auxiliary buds that might be off to the side, like down here, that if for some reason the primary bud gets cut off, the auxiliary can cut in and start growing towards the sunlight as well. If we do this on a house plant, it's called pinching back, and it causes the plant to get fuller, to spread left and right as opposed to top and down, right, where it would only be a top growth for a plant. All right, looking at the breakdown again, remember we have ground, we have dermal, and we have vascular. As you can see here, we have the ground tissue, which is going to be this pinkish area, the dermal, which is on the outside, and the vascular, which are these blue little circles. Since we have a ring formation in this stem, this would be more of a dicot. If you saw a pattern in a stem where you saw ground here and then the vascular being kind of all over the place like this, this would be more of a monocot breakdown. And as you can see, we have xylem and phloem. Xylem is going to be the kind that's on the inside. Phloem is going to be the stuff on the outside. And notice that we have this not only a meristematic tissue at the tips of the buds, but also a ring of meristematic tissue on the inside. That is called the vascular cambium. The vascular cambium is going to lay down xylem and phloem. In consecutive years, we're going to add more and more xylem and more phloem. The phloem keeps pushing outward. The xylem keeps pushing inward. And then we have the what is called the Cork Cambium, which is found on the very, very outside here, which is actually going to make what you consider the bark. The bark actually is the phloem and that dead phloem, which is the bark. But what you call bark, the stuff that sloughs off the tree, is actually the outside dead phloem that has been made via the Cork Cambium. So you can see continual dividing of multiple years that we get this thickening process, and that thickening process is because of the plant laying down more and more xylem inward and more phloem outward. But once again, that phloem falls off, the xylem keeps building inward. So all this stuff on the inside is considered deadwood, and that deadwood is, or heartwood I should say, is dead. That heartwood is not functional anymore. It's the xylem that is functioning near the vascular cambium that's going to be important. Excuse me. Leaf now. Leaf structure is going to be common via, common in both monocots and dicots. We're going to see the dermal layer on the outside here on both top and bottom, which we're going to call the epidermis. On top of the epidermis, we have this cuticle layer, which is waxy, so lipid-based if we're connecting all the way back to biomolecules, and this is going to prevent desiccation, so we're going to slow down water loss. On the bottoms, or they can be on the tops, we also see in this epidermal layer these guard cells, which we'll come back to in a little bit, and the guard cells control the opening of the stomata. And then we have this mesophyll tissue here, which there's two types of mesophyll. There's palisades and there's the spongy mesophyll tissue, and then vascular tissue in the center, so xylem and phloem. So this came all the way up from the roots, and now we've got all these little veins that are going to the plant up in the leaf that are going to distribute and move things. So we might make sugars in here and send it down the phloem, or we might be bringing water up via the xylem to feed these cells so that they can do their processes. So since all this ground tissue, the palisades and the spongy ground tissue, is green, as you can see, it's all made out of cells that is parenchyma cells, or primary cells, because they're going to all be doing photosynthesis. Which makes sense because this is the photosynthetic organ of the plant. And that's pretty much the gist of a leaf. So we're going to continue on here. We're going to look at real cuts of leaves that have been dyed for us. So here, if you want to pause and try to see if you can identify which one's which, that would be good. And then I'll give you the answers here in a second. This is a dicot, and it is a root. The reason I know it's a root, here is the central vascular bundle. You can see the xylem and the phloem here. Here's the dermal tissue around the outside and the ground tissue here. This one is a monocot root. And we see this because here's the stele on the inside. We see those vascular bundles of xylem and phloem here going around in more of a ring shape, but it's all inside this stele. Then we've got the dermal on the outside and the ground in between. Here, this is a dicot stem. We have all this dead pith area here. We have the vascular bundle right here where we have xylem and phloem, and then dermal on the outside, and cortex being that tissue which is considered, I guess you'd call this ground tissue. Oops, ground tissue. This one then is the monocot. Stem. And you can see the vascular bundles here that have the xylem and the phloem all over the place, not really organized in any way, shape, or form. Okay. Continuing on, we need to talk about the types of cells. So parenchyma, and I don't know if these are spelled right. I was trying to throw them up as fast as I could here. So this is photosynthetic or primary, however you want to remember it. And this is going to be where we see all the metabolic processes. So even though I wrote photosynthetic, if it's in the stem might not be doing photosynthetic processes because it might not be green. So the parenchyma cells there, metabolically speaking, would be doing more of a cellular respiration or a storage process than it would be photosynthetic. So keep that in mind. Parenchyma, I like to say, is child-like structure. So it's not secondary cell walls thickened with lignin necessarily. It's for basic structure. So maybe you'd find it in a leaf that still has flex to it. Comparison would be something like cartilage, right, that has flex to it. And then sclerenchyma is going to be more strong, right, aged structure, which you would see in a tree that has grown for many years. You're going to see this strong tissue, more like a bone that is osteocyzed, right, calcified, compared to one is with parenchyma, where it'd be cartilage-like and have a lot of flex to it. Xylem Transportation, vascular tissue. We've got the tracheids and the vessel elements. Remember, tracheids and vessel elements are both dead at maturity. They're going to be transporting water mainly. Tracheids are going to be a little bit larger. Vessel elements are going to be shorter and fatter. Phloem is transporting food. We've got the sieve tube members, which are the primary cells that are doing this, and the companion cells, which are going to help out. They're going to help load things in. Remember, we talk about this is all going to be related as far as movement to transpiration. And this one's going to be all about translocation of sap, right, when we're going source to sink. We'll see that here in a little bit. All right, apical meristems. Apical meristems are growth locations. So all three of these are examples of growth locations. So apical meristems, we're going to see these on tips of the plant, whether it be at the shoot tip or a root tip. It's going to be on the tip. So we're going to see growth upward or downward. Vascular Cambium is going to be laying down the new Xylem and Phloem. Okay. And we're going to see this on the inside, so this is going to give the plant more girth if it continues, and this one's going to be more length. It's going to get more girth if it has consecutive years of growth. So something like a tulip, it's not really going to have this vascular cambium producing multiple layers of cells. It's going to be boom bust, right? It grows one year and it dies back, and then it grows again the next year, brand new again, and dies back. Something that would gain girth would be something like a tree or a bush that has continual growth, consecutive years. And then the Cork Cambium is all going to be making that dead protective outer layer. To the organism that is growing all those consecutive years. All right, transport. We've got cellular transport here, so apoplastic, symplastic, and transmembrane. So in this diagram here, you can kind of see apoplastic is going to be through the cell walls here, that pink line, right? And then we get to this, and we're going to talk about this in a second, that endodermis. And then we also see symplastic, which is actually moving through the plasmodesmatas. Transmembrane is kind of a mixture between the two. So it'll go symplastic, then it'll get to the cell wall and go through the cell walls, and then go symplastic, and then maybe through some cell walls. So it's going to do a mixture of the two. What we see in this diagram here, which is really important, this endodermis, this thing that rings the outside of the vascular cylinder, or the steel where the xylem and phloem is, this is kind of a waxy protective coat that's inside this endodermis, and it's going to guarantee that if something is trying to travel apoplastic, it has to actually go through the cell membrane. So we're ensuring that we're doing a symplastic movement, so we're going through the actual cell, so the plant can actually guide or organize or fend off, or whatever the case may be, govern, I guess, what is moving through the cell and going to get into the vascular system. Because once it gets in the vascular system, it can move now, right? It's no different than in a human. If something is localized, maybe just on your fingertip, say something like a staph infection. If you have a staph infection on your skin, not as bad as if that staph infection gets into your vascular system, starts moving throughout your body. You're going to have more issues in that scenario. So this is the way for the plant to kind of protect itself from things spreading throughout that it maybe didn't want to have spread throughout. We have transpiration that we need to talk about. Transpiration is all the movement of water. The movement of water because of loss on the leaves. So here, water is being lost. We're going to see a negative pressure, so the pressure is going to be low here. Pressure is higher here, so we get a force in of water pulling in through the roots. This is called a photometer. It's a way to set up a lab similar to kind of what we did, but killing the plant here where we kept ours alive. You need to understand how to set up this lab because this is one of the AP labs. So we're measuring water loss because water is being lost from the leaves. So things like humidity are factors. If the humidity is high, that means that the water out here is abundant, so the water loss is going to be low. So we're going to see a low amount because of humidity. If wind is high, if the plant can't counteract and close its stomata, we're going to see a high amount of transpiration. If we see dry conditions, we might see a high amount of transpiration, right, which is the opposite of humidity. If we see a lot of light, because it's doing photosynthesis, it's going to need to exchange gases, so it's going to have a lot of transpiration. And then any other factors which would require the use of water are obviously going to cause a transpiration rate to increase as well. This image is showing you translocation. And translocation is all about sugars being made in photosynthesis, being transported to places where it needs the sugar, so maybe down in a root. This is source to sink, so all these would be the cells that are governing it. So these are sieve tube members and maybe some companion cells, but it's all going to be source to sink. Source can vary. Source in this example is a mature leaf, and the sink might be a root. In the spring, a plant is growing leaves, so it really doesn't have mature leaves to transport sugars, so it's going to need to do some mitosis to grow leaves. So the source might be down here where it stored stuff from the previous summer, so maybe down in a stem or a root, and it'll start sending the sugar upward towards the stems and make new leaves. Right, so just keep in mind that source and sink can flip-flop. Guard cells, you need to understand that guard cells are the cells that govern the opening of the stomata, and the way that they're regulated is through active transport of K+. So if potassium is pumped into the cell, water follows because of osmosis. So if water fills into these cells, the stomata opens. If we pump the K+ out, water follows, and the guard cells become flaccid. And because it becomes flaccid, the cells have an unflexed version, so they actually kind of close together here, and the stomata closes. Okay, so that's all potassium governing, and this is active transport. So the plant has to work to do this. Pen's not working the best. Water potential is a formula that you're going to have to know. So water potential is the psi of W, and as far as we're concerned, we're just going to deal with the pressure in the solute. So the scenario would be like a root and the actual soil around it. If we have something where the soil has a little bit of salt in it, keep in mind fresh water has a solute of 0. So if it has a little bit of salt, this might be a negative 0.1. And maybe the pressure outside is pretty minimal, right? Barometric pressure changes or something. So maybe the pressure is pretty negligible. It might be like a plus, I don't know, 0.2. So overall for this system we have a 0.1 that's a positive. Inside the root we might see a solute concentration, because remember roots do have stuff in them. We might have a solute concentration of 0, negative 0.05. But because it's going through transpiration, there's a pull, so it's a negative pressure, so we might see a positive 1.0. Well, if that's the case, then the overall of the system is what? 0.95. And since we see a positive, I'm sorry, I said a negative pull, not a positive pull. So if we got a pull, because of transpiration, it would be a negative number. So this would be negative, sorry, a negative 1.05. And since this is a bigger number, this is a smaller number, water goes in. The pressure value could be a positive number or a negative number, just depending on what's going on. If we're pulling molecules up, it might be negative. If we're pushing down, it might be positive. Okay, and that is all of the review for morphology and transport. I think the last thing that we should really talk about quickly is tropisms. And that's a plant's ability to respond. So the big one that the AP wants you to know is phototropism. So phototropism is the response to light. So if you have a shoot that's coming up, all right, if light's coming in from this side, what you're going to see later is that you're going to see that shoot leaning towards the light, and the reason is because the auxins all collected on the dark side and cause these cells on the dark side here to elongate and cause the plant to lean. We're going to see this kind of the auxin distribution with things like gravitropism, where it's responding to gravity. So if your cell is here, we'll have these things called stratomyces, which are starch granules in front of the cell. So you have A, B, C, and D. Those starch granules are going to fall down to the bottom of the cell. The auxins will collect down here and cause the cell to grow downward. If we turn the cell so that the cell is now on its side and we have D down here, we have, so it would be C over here, A, and B. Those stratomyces are going to start congregating down here. The auxins will distribute down here and cause the cell to divide and grow downward. So regardless of which way the plant turns, you'll see that response. Pigment tropism is when it's responding to touch. So this would be a plant's ability to close or open its leaves because of something like a Venus flytrap. You know, something comes in. It could be a plant like an ivy that is sending out these tendrils that are swirling around, trying to find something. When it touches onto it, starts growing around it, and then it starts growing up again, and starts swirling around, grabs onto something, starts climbing upward. So that'd be a pigment tropism where it responds to touch. And then hydrotropism, where roots grow towards water sources. All right, so we see this with a lot of sewage pipes in the area getting infiltrated by roots of trees, which can cause backup into your house, and that would just stink. No pun intended. All right, so that is your plant review. Good luck. Let us know if you need any more help with anything.